Swayam Prabha. Digital India. Educated India.
All right, let us get started. See, in the last class, remember we were discussing different kinds of specifications. Autonomous specification leads to what kind of development? Conditional, regulative. Okay, and then the last one we discussed was the syncytial specification. So I told you syncytium means having lot of nuclei in a common cytoplasm, right? One cell having multiple nuclei. So that is the situation in Drosophila embryo. Initially you have nuclear divisions without cytokinesis generating the kind of structures that you see here. And there we talked about specification is based on where the nucleus is, a given nucleus is located, and why that matters is because you have a concentration gradient set up there, right. So you have some components more concentrated in the anterior, and then it slowly decreases as you move towards posterior, and vice versa for some other components, right. This concentration gradient is going to be the topic today. Okay. Some molecules function primarily based on their concentration gradient, and that is what this French flag model tries to explain by Wolpert. So Wolpert has another well known textbook on developmental biology. So the Scott Gilbert is, I feel, is more rather better organized for our purposes, and therefore we follow that, but there is another equally good book that is by Wolpert. So this sort of the same that we learnt in a different context, where how an underlying mesenchyme might instruct an over the epidermis that is over it, right. Like we saw in the skin development, depending on mesenchyme of which region you are transplanting with an epidermis, so then you might either form claws or feathers and so on, right. And then we also saw the newt and frog transplantation, remember the mouth parts being swapped. So this is if you remember that well, you will find this easy to understand. So the key word before even we start is this word morphogen. Okay. So morphogen are molecules whose activity is directly dependent on their concentration. Okay. So at different concentrations they will have different effect. So that is the hallmark of a morphogen. So morphogen will not have the same effect at different concentrations. Okay. So how the gradient matters, how concentration matters, that is what we are going to learn today, and this French flag model of Wolpert is something that tries to explain. So here you assume you have the concentration of a given morphogen is highest here near this area, and then it slowly goes down as you move away on the x axis from the origin. Okay. At different concentrations it is going to make a different specification, let us say different fate for those group of cells. So let us say group of cells that have this concentration or higher is going to make blue pattern in the French flag, let us say this is French flag, right. So highest concentration means blue, and the cells in that morphogen concentration field, let us say this is morphogenetic field, this whole thing, and in this area of the field you have a certain intermediate concentration that is going to make them become white. They are going to be specified to make the white part of the flag. And then when you have very low amount of morphogen, like below this threshold, it is going to make the red. Okay. So now we are going to do transplantation with the American flag. So this is American flag. Okay. So now let us see what is going to happen. So we are going to take a part of it, let us say this part, and we are going to put it here, and then swap. From here also you swap, and the end result is shown here. So now I am going to take cells from here, American flag, okay, and put it in this corner, where it is going to sense the morphogen at its highest concentration, but it is not going to make the blue, this corresponding part of the French flag. Instead, whatever instruction it already has, based on that it is going to make the structure. Like in this embryo, in this part it would have made this stars and blue color background, and that is what it is going to do here. And similarly the opposite, you take this and put it here, it is not going to make this portion. Instead, it senses that gradient, okay, the gradient is this lower level, but then whatever it would have made here, the same group of cells, and that is what it would make here. It is not going to make these horizontal blue and red stripes. Okay. So this is what is French flag model to explain how morphogen gradients work. So again you can go back to the primary, secondary, tertiary fate of vulval differentiation. So you get the highest amount of that the RTK signaling, then it is going to be the primary fate. If you get slightly less, the very same cell, okay, the P6P put at the adjacent P4P position, it is going to have the secondary fate, and away it will be tertiary fate. So the tertiary fate cell, if you brought it to the the highest concentration of signaling from anchor cell, it is going to be primary fate. So that is what you are seeing here. So the cells here, like what it means is all these cells have a certain developmental potential, where given the right instruction they could make this blue color tissue or the white color tissue or the red color tissue, and what determines what is that instruction is the gradient of this morphogen. So highest morphogen means the same cells will make blue, and the cells transplanted to another place like the lowest concentration gradient, it will make the red. This red and blue making instruction is there in the French flag embryo, but that is not there in the American flag embryo, which also responds to the same morphogen gradient, but its genetic programming that has happened in earlier steps is that this is going to make horizontal stripes or the stars and blue color, and that is why this happens. Okay. Reciprocal transplant develop according to their final positions in the donor flag. So this is the idea of morphogen and morphogen gradient. It is really important because it plays a lot of role. This is one place where people did some fruitful mathematical modeling, okay. That is the only one example of using maths in developmental biology, and that is not there in the later editions of Scott Gilbert. So this morphogen keeps coming again and again, okay. So remember this well, the primary thing, if anyone asks what did you learn about morphogen in developmental biology class, you need to tell these are molecules whose activity depends on their concentration. At different concentrations they will have different developmental outcomes. The fate they specify will be different at different different concentrations. Morphogen means you remember concentration specific, developmental specification. You are not relating to anything else, it is actual value. So for example, this is the threshold. So above the threshold, yes, it is a range. So from here to here it is going to be white. So you are not comparing relative to anything else, it is that particular morphogen, its concentration. That pattern is not going to make, the actual concentration will matter. Suppose let us say this is like 100 picograms, let us say 100 picomoles, this is like 1 picomole. Now let us say this is about 50, this is about 20. Now instead of that, I am going to take 1000 picomoles and come to 100 picomoles, there you are not going to see this pattern, all of them are going to be blue, right. So, the absolute value matters in that sense. So, you will see that in an actual example in the next slide. Yeah. So, here is an experiment to show how the morphogen gradient works. Okay. So, this is done on zebrafish embryo. So, this is the surface ectodermal cells. So, here you are injecting nodal mRNA. So, this is one of the morphogens here. So, nodal protein, its gradient matters. Now, laterally if you look at it, it is somewhere on the top, and on the surface you have from the center to one end you have a concentration gradient. Okay. So, now you inject about 4 picogram nodal mRNA into this cell, and the nodal protein produced diffuses across, and when it does that, when it reaches a certain gradient, it is going to induce a certain downstream target like floating head, okay, that is going to be induced, and that is going to become the notochord. And then adjacent cells to the right, so they get much less nodal protein, and they are going to start expressing no tail, and they are going to become the muscles. Okay, no tail is the name of a protein there. All right. So, the one that receives the highest concentration is going to express goosecoid, and that is going to make the head mesoderm. So, if these cells you swap them, then accordingly based on the nodal concentration their fates are going to be swapped as well. So, that is experimentally demonstrated in the subsequent sections here. Like if you have far less nodal mRNA injected in these cells, they directly activate no tail. The concentration is far below what is required to activate goosecoid. These are not difficult to understand. If you want to think chemically, think about the affinity for the interaction between the protein and the DNA sequence. Suppose if something interacts, like enhancer binding happens only at a higher concentration, if the affinity is low, then those enhancers will not be active at low concentration, right. The relative affinities can explain this. You do not actually need to invoke more complex chemical principles here. And instead if you inject a whole lot, like what she was asking, right, is it absolute value or relative value. So, here you are giving really large amount like 6 or 60, does not matter, that 10 times difference does not matter, all of them are going to activate goosecoid. At a high concentration they are all going to respond to it. So, now we are going to do more testing, like controls to ensure that whatever we are interpreting on those experimental results is correct. So, one thing is remember induction competence. So, not any cell is going to respond to the nodal protein. So, if you have a different set of cells that are not going to express the nodal receptor, they are not going to respond to this concentration gradient. Okay, so they lack the competence. So, they are not going to be the right responding thing. So, this is not going to induce any cell. And now, if it is only diffusion of the morphogen and not adjacent cells, like for example, this cell underwent some developmental alteration of its gene expression, and that in turn made this white, that in turn made this green, and that in turn made blue. Then if I replace them with a different kind of cells, then this should become blue or purple. So, that is what is done here. So, you put different kind of cells here. So, they are not interfering with the nodal diffusion, but they themselves do not respond. So, but still these cells, when the concentration comes to the right range, then they become these two, they assume these fates, this green and purple fates. So, the diffusion matters, the intervening cells whatever they are, it does not matter. And if I take a different cell with the appropriate receptor and put it at any position in this field of nodal gradient, then appropriately it should respond, and that is what you see with this cell becoming green. And it is all nodal dependent, not because you pricked a cell with a needle. So, you put laxi mRNA and nothing happens. So, the effect is only with the nodal. So, this is how you do control experiments. Okay. So, this is also research methodology training, okay, in addition to explaining how morphogen gradient works. In the B, the experiment is to test yes, you are right, the neighboring cells are not the cause. Like what I am trying to say is, what if this becoming green was not because of the nodal concentration, whatever concentration perceived by this cell. Instead, nodal induces certain changes here, and in turn this cell induces this cell to be white, and only when this cell becomes white it induces this to be green, and this has nothing to do with the nodal gradient. If this is the case, if I replace these with a different kind of cells, then that should not happen. Like if I have cells with no nodal receptor, then that should not happen, and that is what is being tested in this. Is this clear? This is a very important concept in developmental biology, morphogen and morphogen gradient, and how they activate different set of genes and therefore different fate specifications. Alright, so if we are done, we have actually covered the very basic concepts and principles. Now we are actually going to get into one particular system to understand how that particular development happens, okay, a case study sort of thing. And Scott Gilbert claims, if you read the text, I agree with him. So, nowhere in biology at no time point this sort of an effort or this sort of success or this sort of thoroughness has been achieved, as has been done in understanding Drosophila embryonic body patterning, okay, how the top versus bottom, front versus back, that axis is established. So, this is the best understood model, okay. So, we are going to learn this in detail over next 2-3 classes. To begin with, let us understand the organism a little bit, because when I first learned, I myself found it hard to understand without knowing the anatomy clearly, okay. Having got used to the extremely simple and machine like perfect anatomy of C. elegans, any other organism becomes little difficult, and you need to understand that. So, first and in many lectures they do not go and draw, explain from the beginning to the tissue that you are actually studying, and that does not help you to picture where that particular tissue is. So, therefore, we are going to start with a fly. So, this we all of us know, we have seen them, right, and maybe you have not looked at a fruit fly, but other insects, beetle, etc., have very similar body plan, and you would have seen in some of them head and the thoracic and abdomen regions clearly, and you definitely would have seen 6 legs, right. You may have seen the ones with 4 wings, etc. Drosophila has 2 wings, and therefore, this is a dipteran insect. So, today we are going to look at this region, abdomen, okay. So, this is the enlarged part of its abdomen, its belly, okay. So, thoracic is like your chest portion, and inside that you have 2 structures like this, and this is what we are interested in, and if you take them out they look like this is one and this is one. So, this is an ovary, this is another ovary, and these have these kind of structures. You can 2 of them have been peeled out from that, and like that many of them together form this. Each one of them is an ovariole, and that is shown here big. So, this whole structure is this ovary, and the ovary is made up of many ovarioles, and one of them is shown here in good detail. And in the ovariole, if you go to one end of it like this portion, you will see a structure like this, okay. In that you have these somatic cells, and inside that lies 2 germline stem cells, GSCs, and these divide, and every time they divide, the division is asymmetric. One cell is going to be made closer to this. This area we are going to call the germline stem cell niche, N-I-C-H-E. So, niche is one pronunciation, this need not be the only correct pronunciation, different people say it in very different ways. So, and all of them are accepted. Okay. Similarly, apoptosis, any way you say that is accepted as correct, although the correct one is supposed to be both P's being pronounced. If you are in doubt, leave that word out and say programmed cell death, easiest. So, one cell is going to be formed closest to the niche, the other cell is going to be away from it, and that is enough to start the asymmetry. So, the one that is staying near the niche is going to receive the niche signaling to stay as a stem cell, and the other one that moved away, the other daughter, like when you make two cells, one will be away from the other, okay, and that distance from the niche is enough for it to differentiate and to become a gamete. Okay. So, that signaling, just to recollect our memory, this is not our interest for now, this is the TGF beta signaling. Okay, that remember BMP, all that we learnt, and one of them is decapentaplegic, that is what is this DPP. So, if you get DPP, you are going to be GSC, if you do not get it, then you are going to differentiate to make the oocyte, and that starting cell we call cystoblast. Now, this cystoblast, one of the daughter cells of GSC's division, mitotic division, that is going to undergo 4 mitotic divisions without or rather with incomplete cytokinesis. It is going to have the link. So, that is coming in the next slide, and these are those cells, like this is divided into 2, 4 and 6 and so on, and finally it is going to make 16 cells. So, these 16 interconnected cells we call as a cyst, one that is what is here, and then as they develop they move like this. Okay. So, that each one is a egg chamber. So, this area where this whole thing is happening is the germarium. So, ovary, ovariole, germarium, and from germarium you get these cysts. Cysts are basically one of the daughters of the germline stem cell. After that initial division, this cell undergoes 4 successive mitotic divisions with incomplete cytokinesis, and that is shown in the next slide. Okay. First division, second division, so these two remain together, and then this is going to divide, and see this is going to be connected to this, this is going to be connected to this. These two are not connected, came from the same one, so therefore there is connection, and from this whatever is going to come, that is going to have connection with this, that is 3 and 1, and from similarly 2 divides into another one 4. So, 2 and 4 has the connection. So, 3 and 4 never had a connection, so they are not going to have connection. Okay. So, there is this red color, I will come back to it. Let us first finish the cell division. So, it is going to keep dividing, and then finally you will end up getting this sort of an interconnected set of 16 cells, where if you see these two cells have the maximum connection, they are connected to 1, 2, 3, 4 more cells, while the other one, see this is connected to only 1 and this is connected to 2, and so similarly you see them, and you never see any other 2 cells connected to 4 connections, and the one that has the 4 connection will get the maximum cytoplasmic output from the other cells. Imagine that transcription happens, protein production happens from the nuclei of every one of them, and then they are all moving towards 1, and these 2 will get the maximum. And although in the book it talks about 1 becoming the oocyte, I know from the people who actually did these studies, either one of the 2 can become the oocyte, it need not always be 1. So, and that is finally going to become this big cell which is the oocyte. So, now you see if we go back to germarium, this oogonium is nothing but this cell, that's when this divided into 2, the one that is going to come, the cystoblast, is also called the oogonium. So that one, as it comes, the cell divisions happen and it becomes bigger and bigger, and then finally the oocyte is out of the 16, the one selected to become oocyte, and that now you are going to show in a different color. The cytoplasmic output from all these 15 other nuclei, they are all connected through these things called the ring canals, and they all come and dump the material in the oocyte, and the oocyte is eventually going to grow larger. Okay. So, I feel this understanding is important to really go ahead and understand how the patterning of the oocyte, embryo, etc. happens. So, now a little bit about the red color, which is shown here as yellow. So, this is this microtubule kind of material called the fusome. This fusome expands through the ring canal. So, moving from this to this, the arrow indicates from which cell it is coming, and that sort of spreads through the ring canal all the way, and this is a cross section stained with actin to mark the outside, that is a red color, and this yellow is the fusome. So, that helps in the transport of materials to oocytes. Okay. So, we are not going to look in it has protein called spectrins. So, the fusome structure is made up of these proteins called spectrins. So, they help in the transport. So, other than that, we are not going to worry too much about it. So, all we need to know is this ring canal and the cytoplasmic contents coming to this one selected cell, and the selection is because it has more ring canals, and that is becoming the oocyte. Now, what we are going to focus on is what happens to this oocyte. So, in Drosophila, also in many other insects, the embryogenesis, like for example the symmetric zygote making the asymmetry, like top bottom front back asymmetry, starts from the zygote, but actually in these insects it starts earlier, it starts in the oocyte itself. So, even before the sperm arrives, the oocyte is already setting the house ready for the next generation. So, therefore, we are going to look at how this oocyte forms its top, bottom, front and back. Okay, so, do not get confused, even Gilbert got confused, you will see that when we get into the diagrams. So, we are doing all of this, the early step of asymmetry in the oocyte itself. Okay, so, in contrast, for example, in C. elegans, it does not happen in the oocyte. So, oocyte is totally symmetrical. When sperm enters, the site of sperm entry, this is true even in human, site of sperm entry marks the posterior, and then it leads to a cytoplasmic rearrangement in the embryo in C. elegans. Then the different like for example, when it divides into A, B and P1, and P1 gets the posterior cytoplasm, and therefore its fate is determined by that, and AB gets the anterior cytoplasm, and therefore its fate is determined by that. So, the asymmetric distribution of components within the cytoplasm starts after fertilization in those organisms, but in Drosophila this asymmetry starts even before fertilization. So, do not forget that. So, when we are going to talk about asymmetry, body plan etc. in oocyte, we are not making a mistake, we are actually talking oocyte, we are not wrongly labeling embryo as oocyte. So, now we have a sort of understanding of what this structure is, right. So, now I am going to introduce one set of cells that we did not talk about till now. So, this entire egg chamber was not only surrounded by basement membrane and did not have any other cells, they have somatic cells. Okay, here again I want to make it abundantly clear, these are not somatic cells, these were produced from that one daughter formed by the division of the germline stem cells, right. So, these are all germline origin, while these are somatic cells. So, they were always there, it is just conveniently we ignored in these diagrams, like see they are there. So, they are there, these actin stained ones. So, if you go back, they are there. So, these somatic cells are called the follicle cells. Okay. Do not confuse them with nurse cells. So, these 15 cells that did not get to become the oocyte, they are the nurse cells, because they are going to nurse the developing oocyte. So, these are all connected, although in this diagram it is not obvious, through ring canal to this cell. So, their transcription, translation, output coming from all these 15 nuclei, they are all going here, and this is going to grow. Clear? And at this point I want to state one more information, that these follicle cells are not committed to any particular kind of follicle cells, they are all follicle cells, plain simple follicle cells. Okay. So, now we are going to make the top, bottom, front, back for the oocyte. Okay. No sperm yet. Okay. This is no longer, it is not yet a zygote. So, it so happens that all these outputs coming from all the nerve cells, one of them is a protein called gherkin. Okay, so gherkin mRNA basically. This gherkin mRNA that gets transported into the oocyte, that ends up localizing between the oocyte nucleus and these follicle cells. Okay, essentially these red color dots. Red color dots are not the mRNA, that is the protein. So, the mRNA, gherkin mRNA that gets put in this region. So, in this particular stage, the nucleus ends up being closer to the one end, like closer to the follicle cells than towards the nerve cells. And the gherkin mRNA that comes between these follicle cells in this nucleus through yet unknown mechanism gets translated only here. So, that is actually the starting of everything. This gherkin being made between the nucleus, like oocyte nucleus, and these follicle cells.
Why does the nucleus move towards?
That is how it forms when during these divisions. During the cell divisions, through the orientation of the spindle, the nucleus ends up being closer to the posterior. So, the gherkin protein made here is received by a receptor on these follicles called the torpedo receptors, and that signaling ends up making them terminal follicle cells. Now, these cells are committed. Before this gherkin torpedo signaling, all these follicle cells are equal, but after this signal, these are now they have now specified into a certain specific developmental fate, which is they become terminal follicle cells. Now, they are going to send signals back. Okay, remember the reciprocal induction competence that we have seen, and that actually the signal is unknown, but as a consequence what happens is the microtubules get formed in a certain orientation, and that unknown signaling from these terminal follicle cells brings this protein PAR1, partitioning defect, the C. elegans protein, it is ortholog in Drosophila we are talking about. Remember, you have heard this PAR1, I did explain this. So, we were learning this while we were learning about what is mutagenesis, how do we map, physical map, like genetic map to physical map. In that context, I said one of the ways to confirm the final ORF is by injecting that wild type copy DNA or by injecting antisense RNA, and while mapping one particular gene, they could not rescue with the wild type DNA, instead they put antisense, and as a control they put sense, both had an effect, and that is how they found it, right. And what was the gene that they were mapping? That is PAR1, partitioning defective. Like essentially this group mutagenized, generated mutants that produced dead embryos, and they looked at the dead embryos to find the ones where the embryo, the first division was symmetric. So, partitioning defective. So, that is this PAR1. Okay. So, this PAR1 gets localized due to this terminal follicle cell signaling to the posterior in the oocyte like this. Okay. and that reinforces this microtubule orientation, you know, plus end to the one end. Now we could start calling posterior and anterior. Okay. So you are going to have minus end at one end and plus end at the other end, and as a result on these roads or rails, now you can have RNA and protein moving, and the motor proteins that you know latch on to like Oscar mRNA, they move towards the plus end. Okay, like kinesin moves to the plus and dynein moves to the posterior, and as a result bicoid is carried to the anterior and the Oscar is carried to the posterior, and the Oscar that gets to the posterior gets translated there into Oscar protein. Then that again starts, you know, setting up the posterior cytoplasm called pole plasm, and this bicoid that goes to the anterior determines the anterior. And during all this happens, pay attention to this size change, because nerve cells are continuously providing a lot of material, and as a result the oocyte cytoplasm grows and enlarges. So oocyte really becomes big, and now therefore this asymmetric localization really now is going to make sense, because there is genuinely going to be asymmetry. In a bigger cell, you know, bicoid here to here, there will be a concentration gradient. So similarly Oscar, you will have the opposite gradient. So this is how the front and back of the oocyte gets formed. Okay, so it all starts with gherkin being translated between this nucleus and in the posterior cytoplasm, let us say, like, because you already start asymmetry because of the gherkin translation, and this gherkin signaling the terminal the follicle cells, as a result they become terminal follicle cells, and only these terminal follicle cells produce the signal that organizes these microtubules. Okay. And as a result of those microtubules, you have this bicoid and Oscar asymmetric localization. And one of the things that reinforces this microtubule formation, and also as a result of the terminal follicle signaling, is the PAR protein, PAR1 localizing to the posterior, and that PAR1 localization to the posterior again reinforces this microtubule orientation, and that microtubule orientation is critical for bicoid going to the anterior and Oscar going to the posterior, and the rest of the anterior posterior is a consequence of these two. So in tomorrow's class we will see how the top and bottom are determined. Okay, so we have seen the front and back, so we need to see the asymmetry between these two sides. So that we will do in the next class.